I don't have any disclosures other than I would like to acknowledge the funding from NIH shown here. I'm going to be talking a little bit about history of ME/CFS, which many of you, of course, know about, but I think there's some people who are new to it because of long COVID who are here, and so that's what I will start with. So, actually, an estimated 65 million people are affected worldwide with ME/CFS. This is something that's not cited very often because it's in a peculiar location, but we actually looked at some of the data and think that this estimate is actually more accurate than the other estimates that are out there, many of which are very old. And coincidentally, this is the same number of people who are thought to have long COVID now, but the people who have ME/CFS, of course, have been sick for decades rather than 3 years, many of them. And it has occurred in outbreaks and as sporadic cases, and it's interesting that the outbreaks have sometimes been associated with polio outbreaks. So we have gradual or sudden onset, and unfortunately, no drug and no cure. On the left, you'll see the symptoms developed by the IOM committee to be diagnostic for ME/CFS. But in addition to those symptoms, there are a number of other symptoms that are largely inflammatory symptoms that you might have if you have a viral infection. And in fact, enteroviruses have been most often implicated in these ME outbreaks. Also, in the ME outbreaks, there's been reactivation of herpes viruses observed as early as for example, in the Incline Village outbreak in 1985, there was observations of reactivation of HHV6. So to give a brief history of the ME/CFS outbreaks, you can go back to the 30s and 40s, although actually Byron Hyde has found some that occurred in the early 1900s. Over 75 documented outbreaks in these locations shown on the world map. And then in the 50s and 60s, that was when the name myalgic encephalomyelitis was coined here in London at the Royal Free Hospital outbreak. In the subsequently, additional definitions of ME/CFS, and that's why we're calling it ME/CFS, have been developed by the CDC and the Canadian consensus criteria and also the Institute of Medicine. So I would like to mention that in my thesis is that pre-2020 ME/CFS is a post-viral illness. Most of it is. There may be some cases that aren't, but I believe that most of it is a post-viral illness. A majority of ME patients identify an infection as preceding their long-term condition. So what about those who suspect non-infectious causes? I want to give an example here of some data from COVID-19. This data is from a group of people who were required to have PCR testing whether or not they had symptoms. And what was interesting is how many of them were true asymptomatic. They never had any symptoms despite being PCR positive. There were some in this particular test that were tested and they were pre-symptomatic. Later they got symptoms. But a fairly small percentage were actually symptomatic. So this is happening now. There may be somebody out there who has in fact got COVID-19 and doesn't know it. So likewise, I think my hypothesis is that most ME cases without obvious preceding illness actually arise from asymptomatic viral infections and not from emotional stress, physical injuries or exposures or mold, although these things can be certainly contributing to whether you actually persist in your ME/CFS condition. So there are some lessons for ME/CFS from long COVID. An important lesson is that symptoms of long COVID can arise after an asymptomatic or mild case of COVID-19, just like ME/CFS cases can arise after what some people think is, a little cold, and then they come get ME/CFS, and that sometimes happens long after this little cold. And long COVID, in fact, may appear weeks after the acute infection seems to have ended. And I'm just showing one of the many references about this fact that long COVID can follow a very mild or asymptomatic infection. Now, I'd also like to point out that both enteroviruses and SARS-CoV-2 can cause long-term infections. It's been known for many years. There's a recent paper from 2019, but also an old paper here showing that you can infect human cell lines with, persistently infect them with Coxsackie virus, which is a type of enterovirus. And we're still finding out how long SARS-CoV-2 can infect someone. We don't know if people with long COVID, in fact, have persistent infection in some reservoir we don't know about. But it's certainly possible, and more information about this will become available in coming years. But there have been cases already of people who are immunocompromised who've kept SARS-CoV-2 in their body for over a year. So what are the leading theories of the underlying cause of ME/CFS? I would say there's structural damage affecting neurological function. There are the idea of pathogenic autoantibodies, persistence of abnormal biochemical pathways that I think you'll be hearing about shortly, or gene expression persistently abnormal. Or there's chronic viral infection, and this could be an inciting virus that's still around, or it could be reactivated viruses. Or it could be a combination of these things, so that if you have 2 or 3 or 4 of these, then you persist with your illness. But what I want to point out is that there's that we can't assume that ME/CFS that these things can't all arise from a viral infection, since many of these things can happen after SARS-CoV-2 infection. So the question is that I'd like to address is there any evidence for chronic viral stimulation of the immune system in pre-2020 ME? And by that I mean, could it stimulation by the inciting enterovirus, if there is one, or some other virus, or reactivated herpes viruses, or say activated endogenous retrovirus? I don't know which one of these might be occurring, but the theme is viruses. Any one of these viruses might be doing something. Now, obviously, I wouldn't have asked this question if I didn't think, in fact, the answer was yes. And what I'm going to show you is our evidence suggesting, in fact, that there's chronic stimulation of the immune system. So the evidence I'm going to show you is first about metabolism of immune cells, and then markers for immune cell exhaustion, and then evidence for activation of classical monocytes for migration into tissue. So first of all, the metabolism of immune cells. So T cells alter their metabolism to respond to virus infection. And these there are 2 main types of T cells that I want to mention: CD4 T cells, because they have the marker CD4 on them, and CD8 T cells, which have the marker CD8. The CD8 T cells are the ones of most interest to me. These are ones that cause death. Their role is to cause death of virus-infected cells, and these are in the circulation. CD4 cells are important too, to tell immune cells that there is a danger afoot. Now, T cells become activated when they interact with a dendritic cell that informs them of the presence of peptides from a virus. And after the T cell has been informed about these peptides and that there's a virus present, the T cell can then go and find an infected cell and destroy that infected cell. But in order to be able to destroy the infected cell, the T cell has to be activated by that dendritic cell recognition, or also by some signaling molecules. So when the T cell is being activated, activation on this side here, the metabolism of the T cell is supposed to change. And this is a quiescent T cell. It hasn't been activated, and here's one that it has been activated. And the characteristic is that the quiescent cell is using a lot of fatty acid oxidation to operate its cellular functions, while during activation, glycolysis and oxidative phosphorylation increase. What I'm going to show you is that ME patients have a defect in this activation, the glycolysis and oxidative phosphorylation. And in fact, instead of using fatty acid oxidation here, they are actually using more. Instead of using glycolysis and oxidative phosphorylation in activation, they're using fatty acid oxidation. So glycolysis is low, and so here's late basal glycolysis on the y-axis, and if you compare the ME patients to the control, the ME patients in general are lower. Each one of these dots is a measure of cells from an ME patient. Mitochondrial membrane potential is also lower in the ME patients compared to the controls. And this would mean that their oxidative phosphorylation is probably affected. And then recently we've published that the fatty acid oxidation is actually increased in the ME patients, and this is probably a compensatory mechanism to this lower glycolysis and oxidative phosphorylation. Well, this phenomenon of this abnormal T cell metabolism is actually a phenomenon that is characteristic of T cell exhaustion. So long-term stimulation of the immune system by chronic viral infections is known to cause T cell exhaustion. So here's a dendritic cell being told to stop stimulating this T cell because he's become exhausted by all that stimulation. So if that's the case, it's not enough to just see this abnormal metabolism. You can't conclude you have T cell exhaustion from that. You have to do more experiments. And so we did those more experiments to ask whether CD8 T cells are in an exhausted state in ME patients. So exhausted cells can be identified by the presence of inhibitory receptors and by specific transcription factors. In this part of my talk, I'm presenting work that I did not present yesterday. So this is new. What I'm showing is new to the scientists as well as to you patients and advocates. This has not been even submitted to a journal yet. We have one more experiment we want to do before submitting it. So you're getting results hot off the equipment. So there's an inhibitory receptor called PD-1, and the presence of this PD-1 prevents CD8 T cell activation. So it's a bad thing to have on your T cells. There's also transcription factors and that exhausted T cells are known to express in their nuclei that are characteristic of T cell exhaustion. So let me just give an example of one of these transcription factors and tell you what a transcription factor does. So the protein TOX is an example of one of these transcription factors that drives exhaustion. So TOX will go to regions of the DNA in the chromosomes, and it will then regulate gene expression where it lands on the chromosomes. So TOX regulates which genes are actively producing RNA that encodes proteins. And it can either stimulate the expression of these genes or it can turn it off. And so it turns off some genes and turns on other genes. And when it turns off genes, it can prevent immune response. So there's not enough immune response because of turning off some genes. And it also can turn on inhibitory factors like PD-1. There are similar exhaustion-associated transcription factors also involved, and two of them are TCF1 and TBET. There are other ones as well, but those are the ones we analyzed, so those are the ones I'm particularly mentioning. Now, this work was done that I'm about to describe is the thesis work of a very talented student in my lab, Jessica Maya, who actually just graduated. This was just presented a few weeks ago. And her work, she used all female subjects because of the difficulties of, it's a pretty tricky experiment, some of these experiments, so we decided it would be better to use one sex, but obviously this should be repeated with males. So 15 ME patients, 14 controls. So blood was isolated from the patients, and then cells were separated using magnetic beads So you end up with CD8 T cells, a sample of CD8 T cells. And then these CD8 T cells were put into a flow cytometer. And the important thing to know about a flow cytometer is that you can then ask what cells, by doing fluorescence measurements, what cells have PD-1 on them, what cells have TOX, TCF1, or TBET. So it tells you the proportion of cells that have these different markers on them. So the first thing she did was to look at total T cells. So there's lots of different types of CD8 T cells. They're undergoing development. She looked at how many cells have both PD-1 and TCF1. So these are the percent of the total CD8 T cells that have these exhaustion markers. And you can see that the ME patients have more than the controls. And then if you look at a combination of PD-1 along with TOX and TBET, again, the patients have more, there are more cells in the patients with these markers than the controls. And then the combination of all of these, again, more patients have these markers on their CD8 T cells than the controls. But you can also not only just look at total, you can look at particular types of T cells. And so she looked at these two particular types, naive T cells that are early and very early here. And again, they had more TOX transcription factor than the controls. And then she examined these double positive CD8, CD4 cells. These are unusual type of CD8 cells that have been seen in chronic viral illnesses. And again, you can see a dramatic difference between the patients and the controls in the amount of these cells that have TOX on them. And then another, just to show another, she has a lot more data. I don't have time to show it, but I'm going to show this early. If you look at the early CD8 cells, you'll also see a big difference between the percent of cells that have TOX in the patients versus the controls, and also a higher percentage of cells in the patients than the controls have these other markers as well. Looking at the terminal CD8 cells, again, we have the same theme. There's more cells that have TOX than the healthy controls. So what's interesting is this T cell exhaustion is also being considered in long COVID. And this diagram I have here is a paper reviewing what's going on in long COVID. And starting with SARS-CoV-2, activated immune response, you can then end up with an exhausted immune response. And that exhausted immune response can result in latent virus reactivation because your body loses control over those viruses that you've been suppressing all these years. And so we don't know really whether this is happening and there's actually still some virus around in ME patients, or as a result of this, you've activated your latent viruses and that's causing persistence of the illness. So what other indication of chronic viral infection can you get from immune cell analysis? And here I want to present some work from my collaborator, Andrew Grimson. His lab is primarily responsible for what I'm about to describe to you. And these particular individuals here that are in bold are the ones who did most of the work that I'm about to describe. So So immune cells were isolated from blood, and they were put into this device, which is pretty amazing invention, in which cells come along through a microfluidic device, and they get combined with oil droplets. And this combination of the oil droplet and the cell allows you to sequence the reagents there allow you to sequence the RNAs that are present in each cell, so you can get the gene expression that occurs in each one of those cells. So individually, you know what's being expressed in those cells. And the other thing that you can do is that you can use this single cell sequencing data to identify the type of each cell whose RNAs were sequenced. So you can identify which cells are monocytes, which are natural killer cells, which are B cells, which are T cells, and all of those other types of cells that are in your population of immune cells. And you can do that because these different types of cells have characteristic gene expression so that you can see, what they are, you can identify what type they are. Well, when Andrew's group analyzed these single cell sequencing data, what they found is that the type of cell that with the most gene expression differences between ME/CFS and controls were a type called classical monocytes. So in ME/CFS and controls, there was the most difference in their gene expression. And what did that difference mean? Well, if you look at the monocytes, they were expressing the genes indicating in ME they are being signaled to develop into macrophages. Okay, so you have a monocyte in the circulation, it gets signals, it goes through your epithelium, your endothelium here, and gets into the tissue, becomes a macrophage, and those macrophages' job is to go and get pathogens that may be in the tissue. So it seems to be, there's some signaling going on that's telling these monocytes to become macrophages. The gene expression data from the monocytes then was compared by a very talented student, Hongyu Zhu, to using a computational method, machine learning, that I think you heard about. And by doing this, each cell could be classified from its RNA sequences, whether it was normal or diseased. I'm talking about the classical monocytes now. So looking at the data for the classical monocyte gene expression, was that gene expression normal or was that cell diseased? Were you just using that expression? And this diagram is not important to know exactly how this is shown, but this is one case and this is one control. And each of these dots represents a cell, and if it looked normal, it's colored yellow. If it looks abnormal, it's colored blue. So the controls have a few blue cells. They don't, looking abnormal. But you can see this particular case has a lot more of these abnormal-looking cells. And in fact, if you graph this, you can see that the percent of classical monocytes predicted to be diseased was higher in most ME/CFS subjects than in controls. And that's seen here. These are the controls. The percent of diseased classical monocytes is very low in the controls, but there's a range in the cases of the number of these predicted diseased classical monocytes. But one thing that gave us confidence this has some reality with regard to the illness, to ME/CFS, is that when you graph the number of these, the percent of the diseased cells versus the fatigue scale, the MFI-20, how fatigued you are more fatigued if you have more of these diseased classical monocytes. So that gives us some feeling that this, in fact, is a result that's important and could tell you that the classical monocytes abnormalities are having an effect on your condition. So to summarize, immune abnormalities distinguish patients and sedentary control subjects. There's reduced glycolysis and other abnormalities in immune cell metabolism. The expression of this inhibitory receptor PD-1 and exhaustion-driving transcription factors gives more evidence that T-cell exhaustion is occurring in ME/CFS subjects. And the classical monocytes in the subjects appear to have received some signals that will induce them to enter the tissue and become macrophages. And they're there to battle against what's an unknown antagonist that we wish we knew what that was. These are my acknowledgments. And I'd like to give special credit to Jessica Maya, who did not only the T-cell exhaustion work, but she drew some of these really beautiful diagrams, allowing me to explain things to you. Alex Mandarano, who did a lot of the T-cell metabolism, is now doing a postdoc. Andrew Grimson's lab that did the work on the single-cell RNA sequencing is here. I'd like to acknowledge all of my support. And you can learn more by going to our website, where we have a description of what our new renewed center is going to be doing, as well as many of the presentations that we have. And I apologize for, there seems to be some incompatibility between my PC and the one up there. So some of the fonts were a little weird, but otherwise, thank you for your attention.
Thank you, Maureen. Questions? Any questions from the audience? Richard? Has Richard got the cube? Oh, here. It's on its way.
Thank you. I was wondering if you have any thoughts about maybe the use of statins to stop the monocytes from macrophaging.
Yeah, there are a number of interesting things that one could use to potentially treat these problems. For example, for so PD-1 and these transcription factors, they're also present in cancer. So cancer is stimulating your immune system all the time. So a whole wealth of drugs have been created to turn off T-cell exhaustion. And yes, there's certainly a possibility that there are a number of drugs that could deal with the classical monocytes as well as the T-cell exhaustion. But I'd like to comment that those drugs are not something to be trifled with, that we really need, even the statins are not fun to take. And so we really need to have clinical trials. I really feel there's dozens of drugs we should be trying right now in clinical trials in ME, and that's some of them.
I was just wondering if there's any explanation, given the work you've just presented, with why, if one actually stays within one's energy envelope, even though you feel crap all the time, it's one level of illness, but then there's a whole different level of illness if you actually overdo it, and then there's the crash, the PEM, which is just like, it's just something completely different, and how that actually correlates with what you've shown?
Yeah, that's something that we obviously would like to know, and in fact in another study that I didn't present, we actually make people exercise so that we can see what happens after you exercise. One possibility, this doesn't explain when you do something very minor, like and people can crash, I know, from very minor things. But for people who overexert themselves by doing something physical, one thing happens when you exercise your muscles is there's inflammation that occurs, that molecules get released. And so that extra inflammation from doing any sort of exercise potentially could be making your immune system even more dysregulated. But I don't have a good explanation why someone who is bedridden gets up and takes a shower and then they have this problem. That is probably something neurological rather than immunological.
Okay, thank you very much. So I think we'll stop there. Thank you again, Maureen. Any more questions for Maureen? She'll be outside the coffee area. I'd just like to thank the three speakers we've had in this session, Alain, Nancy and Maureen. Thank you.